I'm very excited that we have Dr. Melissa Ilardo here today. So she did her bachelor's degree at Princeton in ecology and evolution, and then did a master's in microbiology in Hawaii, and then moved to Copenhagen to do her PhD. And as we were talking, things changed a little bit. So maybe started with the intention of working on ancient DNA, but then transitioned to modern human population genetics, which I find amazingly fascinating, and obviously you all do too because you're here. She's now doing a postdoc at University of Utah and UC Berkeley, and today is going to talk to us about adaptations to diving in sea nomads. Thank you for coming.
So I think, is this on? I'm all set? Okay. Well, thank you so much for coming. As Melissa said, I'm going to talk to you today about a population called the sea. Oh, sorry. Yeah, sure.
Can you move the microphone up a little bit so it's
Up. Is that better? I'll also just try to be louder. Is that okay? Yeah, so I'm going to talk to you about a population called the Sea Nomads. And they're called the Sea Nomads because, well, first let me say that the Sea Nomads refers collectively to a number of culturally and linguistically distinct populations throughout Southeast Asia. So you have the Moken, Moklin, and Urak Lawoi in Thailand and Myanmar, the Orang Sukulaut in the Riau archipelago of Indonesia and around Singapore, and the Orang Bajo in the Philippines, Malaysia, and Indonesia. And most of these names just mean sea people in their respective languages because that's how they're known within the cultures where they live. So they're called sea nomads because traditionally they spend their whole lives at sea, living in houseboats like these, traveling around and only visiting land occasionally. They spend so much time on the water that it's often said that sea nomad children learn to swim before they learn to walk. They have a really deep connection with the sea. And so this is a Moken village in Thailand. And this photo was taken by a Thai anthropologist in 2003. And I point out the year because in the 2004 tsunami, this entire village was devastated. It was destroyed. There was nothing left. And yet not a single Moken was killed in that incident. And in addition to surviving, they were actually able to save the lives of others, including some tourists who were in the area, by reading and reacting to the warning signs that the tsunami was coming. So they were able to take their boats to deeper water and on land to move to higher land and survive. They've existed for thousands of years as marine hunter-gatherers, and they do this through a combination of fishing from the surface, but also through free diving or breath hold diving. And they do that in order to spear fish, like you see here, with very minimal equipment, but also to collect food such as mollusks and sea cucumbers, which make up a big part of their diet. And they're extremely good at this. So not only can they hold their breath for impressively long periods, there's not really good data on how long exactly they can hold their breath. I've been told 13 minutes by a diver. I'm not sure if I believe that. I didn't see it myself, but I have seen them dive for very long periods of time. But in addition to these very long dives, the time between their dives is extremely short. So they're actually spending so much time underwater that it's on the order of marine mammals like the sea otter. So they're spending about 60% of their working day underwater. So if we compare them to other diving populations, here you have I wonder if I can point at this. No, okay. So the Ama are a group of pearl divers in Japan, known for their diving ability. And then we have the Bajo over there. And these were observations taken during a normal day of diving. So this wasn't anything special. This is just, I believe it was 10 consecutive dives. And you can see that the Ama are spending about the same amount of time underwater as they're spending at the surface recovering from their dives, whereas the Bajo, as I said before, are spending about 60% of their time underwater. So they're spending more time underwater than at the surface recovering. And this makes them extremely unique. Free diving can be extremely dangerous. And the reason that it's dangerous is because when you dive, or when you hold your breath for a long time, what signals that you want to breathe again isn't actually dangerously low oxygen, but it's the buildup of carbon dioxide. So long before you need oxygen, your body starts telling you to breathe because of this carbon dioxide buildup. So really experienced free divers will train their bodies to overcome this initial signal. But now that you've overcome that signal, there's nothing actually telling you when you need to breathe. So even experienced free divers will sometimes drown because they lose consciousness as they're ascending. So while this isn't so good for the divers, it's extremely good in terms of studying natural selection, because here we have a strong selective pressure where if you're not good at diving and you're doing it all the time, there's a good chance that it could kill you. So in order for natural selection to act, it has to have something physiologically upon which to act. And so that's when I came across the human dive response. So the human dive response is actually triggered by the combination of holding your breath and submersing yourself in cold water, or water that's cooler than the temperature of the air. And you actually don't even have to submerge your whole body. It's just there's something about submersing your face, and there's a nerve stimulation that's involved, which causes this human dive response to occur. And so what happens is, first you have bradycardia, so your heart rate slows down to preserve oxygen. Peripheral vasoconstriction, so the blood vessels in your fingers and toes get smaller in order to preserve that oxygen-rich blood for the organs that need it the most, because the chances are your fingers and toes will be fine while you're diving, but your heart and brain and lungs really need that oxygen. And then lastly, contraction of the spleen. So this is kind of a weird one. I don't know about you, but when I think about diving, the spleen is not the first organ that I thought of. In fact, you can live without a spleen. So how important could it really be? Well, first I'll tell you a little bit about what the spleen does. The spleen filters blood. It's involved in the immune response to certain bacteria, and it stores red blood cells, which is how it comes into play with the human dive response. So when you dive, as I said before, your spleen contracts, and it pushes this reserve of red blood cells into your system, giving you a 10 to 15% increase in terms of available oxygen. So you can think of it kind of as a biological scuba tank. And there hasn't been that much work done on the spleen in humans, in particular in response to, or as it relates to the dive response. But we do know from comparative physiology that certain species of seal, like this is the Weddell seal, and in addition to being really cute, these seals are exceptionally good at holding their breath. So even compared to other species of diving seals, these seals they have a very high dive capacity. So they can hold their breath and dive for a very long time. And they also have disproportionately large spleens. So the Weddell seal's spleen takes up almost half of their abdomen, which makes them unique amongst diving seal species. So I thought that if we find this to be true in seals, where natural selection seems to have acted to increase spleen size in order to increase dive capacity, then we might find the same thing to be true in humans, where natural selection has acted in sea nomad populations to increase the size of their spleen. So in order to investigate this, I went to a village called Jayabukti. It's a village of about 5,000 people in central Sulawesi, Indonesia. They're living on a small coral island. And most of the people who are living there identify ethnically as Bajo, including all of the people that you see pictured here. Some of them still engage in traditional breath hold diving, including this father-son pair here, and the guy on the right there who is engaging in the traditional Indonesian pre-dive ritual of smoking a cigarette. Something like 75% of Indonesian men smoke, so smoking is extremely common in these populations. And as a comparison, I went to a village that's about 20 kilometers away called Koyoan. And this village is populated by people who identify ethnically as Salawan. And so Salawan is different from the Bajo. And even though the Salawan are living quite close to the sea, you can see, I mean, it's just there in the background, they're engaging with it very little. And that's because the Salawan have existed historically as farmers. So you can see here they have cows, there's some goats, in that photo over there, there's chickens. So whereas the Bajo children are in the water basically from the moment they're born, most of the children in this village and most of the adults didn't even know how to swim in spite of having it right there. Same water, it's not like the conditions are different. So it's very accessible to them, but they're not diving or interacting with the sea. So from each village and each individual, I took DNA samples in the form of spit samples, which we processed in Indonesia. And I also brought with me a portable ultrasound machine. And using this machine, I was able to measure spleen sizes. So here I am in the home of one of the divers that I showed you earlier, measuring the size of his spleen. And what we get are these images. If you can see, that's a spleen, same image. And by taking it in two planes, we can calculate a spleen volume. And so this is comparable to what we would get from something like a CT scan in terms of accuracy. So we calculated the spleen volumes for all of these individuals. And our first question was, do the Bajo have bigger spleens compared to the Salawan? And so the data from that is here. And you can see both visually, we have the Bajo in blue and the Salawan in red. Both visually and also from that p-value down there, the answer was yes. They have statistically significantly larger spleens. And on average, they're about 50% larger. So a normal spleen is about the size of a fist. So the Bajo spleens would be, again, about 50% larger than that. So this was a really dramatic and exciting result. But we still didn't know whether this increase in spleen size has something to do with the actual act of diving. So is there something about diving that makes your spleen bigger? Or can you expand your spleen? Now, this is a question that even 100 years ago we would not have been able to answer. And that's because the Bajo, traditionally, everybody dives. Men, women, children, everyone is diving. That's the only way that they're making their living and surviving. However, now that these populations have become more settled and started to move to more of a land-based existence, not as many people are using the traditional, or living the traditional lifestyle of diving. So of the people that we interacted with, only about 50% of them are diving, which allowed us to compare Bajo divers with Bajo non-divers. So what we would expect is that if there is a genetic basis to this, there should be no difference in spleen size. Regardless of whether or not you're diving, if you're Bajo, you should have this large spleen. And that was, in fact, what we found to be true. So here we have the Bajo divers on the right and non-divers on the left. And as you can see, again, visually, but also from the p-value, there was no difference in the sizes of their spleens. So this, to us, indicated that this large spleen phenotype that we're seeing might have a genetic basis. So in order to identify that genetic component, we wanted to perform a selection scan. Now, you might be familiar with the population branch statistics method of looking for signals of natural selection. And this was used in Tibetans to identify their adaptation to high altitudes. But unfortunately, this method assumes no admixture. So when you're working with a population of nomads, these populations have historically interacted with all different kinds of other populations. And so a lot of admixture has occurred. I'm assuming all of you know what admixture is. It's just when two previously isolated populations interact. So again, with nomads, that's something that you're seeing historically all the time. So we really had to take admixture into account in order to identify natural selection happening in this population. So we instead used a method called cell scan, which actually uses admixture to identify selection. So here you have an admixture graph. How many of you know what admixture is or have heard of it before? Or I should say, who doesn't know what admixture? Well, maybe that's an embarrassment. Okay, so here, this is an admixture graph, and I'm just going to talk you through it real quick. But we have each genetic ancestry or background represented roughly by a color. So we have the Bajo roughly in red on top, the Salawan in yellow, and these are the Han Chinese taken from the 1,000 Genomes Project. And so what you can see is that this individual, like myself and like many other Americans, is admixed. So my father is 100% Italian, and my mom is a mixture of Northern Europeans. So I could be considered an admixed individual because I have some of that Italian ancestry coming in as well as some of the Northern European ancestry. So this individual seems to have some kind of East Asian ancestry coming in, as well as a little bit of that Bajo ancestry and the Salawan ancestry as well. So that's admixture. So what this method does is it looks across the genome and it develops this global estimate of admixture proportions for each of these individuals. And it looks for deviations from that as evidence that natural selection has taken place. So from that, we were able to looked at the top 25 hits from this selection scan. So the top places in the genome where we think natural selection might have occurred. And looking at each of these sites, we then calculated an association. Performed an association test, which asks for each of these sites in the genome, how much does the observed genotype correspond statistically with the observed phenotype, or large spleen? So are you more likely to have a large spleen if you have a particular allele at this site? And from that, one in particular stood out, which was a SNP in a gene called PDE10A. So here you can see that association, again, where spleen size seems to increase the more copies of this derived allele that an individual has. And what is PDE10A? Well, this is a long sentence, but it encodes a cyclic nucleotide phosphodiesterase that helps to regulate the concentration of cyclic nucleotides, thereby playing a pivotal role in signal transduction. I'll get back to what that means in a second. But first, just to show you, here we have this nice overlapping peak. So the selection scan, yeah, so we can see that that site has been under selection. We can see that it's associated with spleen size. And here we have, it also corresponds with a peak in expression, specifically in the thyroid. And I'll come back to that in just a second. But yeah, just to say again, the strongest association for our top SNP is with expression in the thyroid gland, which is an organ that has cyclic AMP-mediated hormone release. Okay, so what does that mean? Well, this is the thyroid hormone synthesis pathway, or representation of it. So here in blue, we have a thyroid cell. And thyroid cells form these follicles. So on the outside, you have blood flow. On the inside, you have the interior of this follicle. And if you follow the arrows kind of in and out of this cell, that's the pathway of thyroid hormone synthesis. And where our phosphodiesterase comes in is up here, so early on in the pathway. And what we see is that the phosphodiesterase seems to influence how much thyroid hormones you produce. And so specifically, what we see in the BAJO is lower PDE10A expression, which corresponds to higher thyroid hormone levels. And although we didn't actually measure thyroid hormone levels in this population because we only took spit samples rather than blood, we did follow up in a European cohort that had thyroid hormone levels, and we can confirm this to be true. So if you have the allele that the BAJO seem to have at higher frequency, you have higher thyroid hormone levels. So that's interesting, but what does the thyroid have to do with the spleen? Well, it turns out if you take mice and you genetically alter them so that they produce extremely low levels of thyroid hormones, you see this drastic reduction in spleen size. So here we have a normal mouse spleen compared to this knockout mouse spleen. So this is a mouse that has really low levels of thyroid hormones and a very small spleen. Interestingly, if you take those same mice and inject them with a thyroid hormone, T4, their spleen returns, it regains size. So here we have, again, control, knockout, and knockout plus an injection. What's interesting about this is that recovery of size that you see there happened over only 48 hours. So it seems to be there's something that the thyroid hormones have to do with the spleen that we're dramatically able to increase the size of the spleen. So we believe that the most likely mode of action is increased spleen size as a consequence of increased thyroid hormone synthesis. And that was the main story of our paper, which came out in Cell. And it got people curious enough about spleens to start Googling them. So this is the Google trends for spleens. When the paper came out, people were like, what is a spleen? Also made it to The Onion, which I was excited about. Anyway, I just want to tell you briefly about a couple other hits from the selection scan. So these are things that were found to be under selection in the BAJO, but don't seem to have to do with the spleen, but do potentially have something to do with diving. So one is this SNP in a gene called CACNA1A, which is activated in response to hypoxic conditions. So hypoxia is low oxygen. That's what you experience when you're diving. And it seems like something is going on here where they've had selection on something in responding to hypoxia. But actually the top hit of our selection scan was it was a SNP that was found just upstream of a gene called BDKRB2, which is actually the only other gene that's been shown to be associated with the human dive response. And this gene specifically acts on that peripheral vasoconstriction component that I talked to you about earlier. So it seems like there's something going on that's allowing the BAJO potentially to increase the amount of peripheral vasoconstriction that they're able to do while they dive to preserve even more of that oxygen-rich blood for them and prolong their dive time. If any of these jump out at you for any particular reason, please talk to me after, because you never know. Sometimes these connections aren't always very obvious, especially like our phosphodiesterase. But so who cares? Well, as I said before, hypoxia, or low oxygen, is a really important issue in a lot of different medical contexts. So from conditions like COPD to apnea during surgeries or critical care situations, it's something that's very relevant for clinicians. And most of the research that's been done on hypoxia tolerance in human populations thus far has been done in high-altitude populations, like the Tibetans. So this is from one of the many famous papers about the adaptation to altitude in Tibetans. But one important distinction is that Tibetans and other high-altitude populations experience chronic hypoxia. So they're experiencing constant lower levels of oxygen. And what we see in the Bajo is acute hypoxia. So there's just a sudden cutoff in oxygen. And so while this might be more relevant for conditions like chronic smokers who are experiencing these same constant lower levels of oxygen, what we see in the Bajo might be able to tell us a lot more about what we see in these critical care situations. Another reason I care about it is because it has the potential to teach us something about natural selection and the evolution of our species in a really unique way. So a lot of times when we see adaptation in modern humans, it kind of tends to be a return to equilibrium. So humans move into a new environment or adopt a new lifestyle that causes some kind of disruption to a physiological process, and then they evolve an adaptation that allows them to circumvent that and return to normal. So an example, again with altitudes, when humans go to altitude, including all of us, what happens is it triggers a response where we start to produce more red blood cells. And this is great in terms of increasing our oxygen capacity, but having a high concentration of red blood cells also comes with a number of health issues, which actually is something that's seen in a lot of professional cyclists who do blood doping to increase the number of red blood cells that they have. So some of these individuals, at the peak of blood doping, before it got a little more under control, they couldn't even sleep through the night because their blood becomes so viscous because they're putting so many red blood cells into their system that if they stay still for that long, they'll die. So a high amount of red blood cells can actually have a lot of serious health effects. So what we see in the Tibetans is an adaptation that blunts this response. So they don't produce the high amounts of red blood cells that you and I would produce when we go to altitudes. So what we see in the Bajo, instead of being a return to normal, seems to be enhanced function, which makes it a very unique chance to study natural selection in our species. What's next? So right now I'm in Utah, and I'm working in a lab that does a lot of work with mouse models and cell lines and other molecular biology techniques. And what we're doing is trying to see, can we recreate what we see in the Bajo in mice? Can we make their spleens big? And so we're doing this currently by injecting them with an inhibitor that inhibits the phosphodiesterase that I told you about earlier to mimic what we see in the Bajo. And we wanted to see what was going on with their spleens at various time points. And so in order to do that, now I can not only give ultrasounds to humans, but also to mice. So this is what it looks like when you give an ultrasound to a mouse. It looks kind of traumatic for that mouse over there, but that mouse is high on isoflurane, so he's very happy. But yeah, you can see that we have very small probes to look at their very small spleens. And so we were able to do this in mice. And we found that after just 4 weeks of treatment with this inhibitor, you can see a significant increase in spleen size. So here we have spleen area corrected for body mass, and in the control and treated mice, and you can see it's statistically significantly larger spleens. We were actually, let me first show you, yes. So what's interesting was that, that's what I showed you before. This was, initially, there's no difference. At week 4, we see a difference. However, by week 9, that difference is there, but it's no longer significant. So we're trying to figure out what exactly happens between those two time points. But after week 9, we actually just last week sacrificed the mice, and we were able to measure their spleens directly. And we can see that if you look at the length of the spleen corrected for body mass, we once again see a statistically significant increase in spleen size in these mice. So what we're doing to follow up on this now is looking at using flow cytometry to determine the cellular composition of the spleens of the control mice and the treated mice to see what's going on at the cellular level in these spleens. So are they just holding more red blood cells, or is there something changing in terms of the composition of the spleens themselves? We're also looking at thyroid cell lines. So these are rat thyroid cells. And we can use CRISPR-Cas9 to genetically modify these cells to knock out that PDE10A gene and try to figure out if, in doing so, we increase thyroglobulin production, which is a thyroid hormone precursor. I'd also like to go back to visit these populations because there are a lot of measurements that I wish I had taken that we can now take, and a lot of new ideas, including investigating that bradykinin receptor, BDKRB2, I told you about, to see if something has changed in terms of their peripheral vasoconstrictive response. What exactly has changed? Is it constricting faster? Is it constricting more? And so we'd like to go back to these communities and take more samples and investigate more questions. And that's actually, I'm a little bit under time, but I would love to take questions from you guys. So with that, I'll just say thank you to all the funding bodies, to all my collaborators, and most of all to the people who participated in my research.
Great.
Sorry, so we'll take a few questions since we do have a bit of time, but then we're not going to keep her on trial, and we'll go to tea a little bit early. So a few questions.
Sorry. I actually have kind of two questions. Like, first of all, for the spleens, could one medical application be to inject a person with higher thyroid to increase their spleen sizes, like before they go into surgery, so less risk of medical complications?
That's certainly something that long-term could be interesting. First, we want to figure out what exactly is changing in the spleens and how quickly that's changing. And there are a lot of things to figure out there, but certainly those are the kinds of things that we're thinking about. And every time I talk about this with clinicians, they have different ideas of ways this could be applied. So.
And also for the Sea Nomads themselves, like I actually met them in a documentary, and it says like they spend so much time diving that their lenses can actually change when they're underwater, so they can see without the use of goggles. Is that like accurate? And do you think that's one of those SNPs that might be more associated, or are we off the track here?
So that was actually one of the first things that I read about the Sea Nomads when I heard about them, because that study was done in the Moken, and there was actually just this researcher. Sometimes I start off talking about this because I think it's interesting. She was on vacation in Thailand, and just a physiologist just happened to notice that the Sea Nomad children could see better underwater with no goggles. And so she actually tested it like right there while she was on vacation against European children who were also on vacation, and found that they had significantly better underwater vision. And she actually went on to repeat the study with only European children when she went home, I guess. And she found that it was something that could be trained in children. And so it was kind of dismissed as not having a genetic component because it's something where you can actually physically change your lenses by repeatedly diving. But the same line of reasoning was used to dismiss there being anything special about the Bajo at all. I actually heard a Swedish physiologist who said, I can train my European divers to dive just as well as the Bajo. There's nothing special about them. So I think it's really dangerous to say just because you can train something, that doesn't necessarily mean that this population isn't starting out with some kind of advantage. So it's something I would absolutely be interested in looking into. And actually at one of my talks, one of the genes that had a SNP, the underselection, someone said had something to do with vision. So it's something that I'm looking into as well, yeah.
Thanks for that talk. That was really interesting.
Okay.
I was wondering if you know anything about the parasite population that these populations are exposed to. And then particularly in the case of malaria, I don't know if there's malaria there, but I know there's in some other populations there's selection for larger spleens in response to malaria infections, and the spleen size is plays a big component in your immunity against malaria infection. So does this have any secondary effects?
I'd be really curious if you could point me to those articles. I haven't actually seen those. The Sulawesi, where they're living, there's not actually a lot of malaria, but down in Flores, where there's also a really big Bajo population, and a lot of other areas where they're living and have historically lived, there is a high amount of malaria. So, I mean, that would be very interesting to look into, yeah. Yeah.
Hi, great talk.
Thank you.
What diseases do these people get? What do they die of? What sort of things do they get? Morbidities and mortalities.
You know, that's a great question. I'm not really sure. I would love to know more about that. There's just not a lot of data on these people. I did meet a lot of very elderly Bajo who were very fit and healthy. We had I think an 86-year-old diver who was part of our study. And we set up beforehand, we said, okay, to be included as a diver, you have to have been diving within the last week. You have to have been diving always. You know, we had a strict set of criteria, and everybody who was a diver met all of those criteria. It was so he had been diving earlier that morning. So when they're extremely active and fit people. I didn't notice or hear about any particular diseases. I know that in Malaysia, the similar populations often have a high incidence of diabetes, but I think that could have to do more with the sedentarization and socioeconomic issues, because they are very much like the Roma in Europe, where they're ostracized and kind of held at a lower socioeconomic status. So, yes.
So, really great talk. The variant that you identified, you said you went back into a European population and found that it was associated with the thyroid hormone.
Yes.
I'm wondering, did you also look and see if it was associated with with spleen size in that population?
We did, and it was like p-value of 0.06, so it was just on that border. But the problem is that it's almost impossible to find a genetic cohort that also has spleen sizes. So that was actually our European cohort that we used. It was from an obesity study where they had happened to take abdominal CT scans. And so someone went back through and was measuring spleen sizes. So the accuracy of those measurements, I'm not sure, but even then we were able to find an almost significant association with spleen size, which was very encouraging as well.
I was curious, so your study's obviously predicated on the existence of a diving reflex or diving response in humans, yet it seems likely that most humans don't live in places where they do much diving at all. So what are your thoughts about the evolutionary origins and maintenance of the diving response in humans?
So it is something that you can measure in everyone. It's not just in these populations. We're actually working on a study with some physiologists in Copenhagen where we measured it in a bunch of Danish students, and we're going to use that as a comparison. But it's something it's universal. It seems to be, and it's not just in humans. It's actually in a lot of different mammal species. So I know I've heard this aquatic ape hypothesis, which I'm not as on board with, because we see the same thing across mammals. So you see it in, obviously diving mammals like dolphins, whales, seals, but also horses have, I'm not sure in terms of whether they've done the diving response, but in terms of exercise, you see a lot of the same things, where when you undergo acute bouts of exercise, you have that contraction of the spleen. And I think also, well, I guess not bradycardia, but so it seems to be across a lot of different mammals, so I don't support, not that you suggested it, but the aquatic ape hypothesis. But yeah, in terms of the origin, I'm not sure.
What about maintenance? Why is it maintained?
Yeah, I'm not sure. That's a very good question. Yeah, maybe we've had enough. I was just learning yesterday, although I think he's not here, but about, I guess there's a population of macaques, long-tailed macaques that dive. So maybe there's just been a lot of diving in the history of mammals and primates, and yeah. Yes.
Hi, this is really interesting, so
Thank you.
thank you. Just a quick question. At the beginning of your talk, you had mentioned that a large percentage of this population smokes.
Mm-hmm.
I don't know if there's any, possibility of a confounding factor from smoking, because smoking is associated with inflammation. And there can be increased spleen size depending on sort of the amount that you smoke.
Right.
So I was just wondering what your thoughts are on that.
So fortunately, the Salawan also smoke. It's just an Indonesia-wide thing where for some reason, Indonesian men, it's the highest smoking rate in the world. And so actually, yeah, it's interesting because almost none of the women smoke. Actually, the only women that we had in the whole study who smoked were our female divers. But yeah, so I think the fact that we have that in both populations controls for that, because otherwise it would be, I mean, it was something that we recorded for that reason, and then we just realized basically everybody smoked. So yeah. Yeah.
Thank you for the talk. I just have two questions about, because I have using the same methods for selection, and one of the things is that what happened with the other signals that even have more high values in the selection scans. Maybe what is the evolutionary explanation for the other signals in the other genes, and maybe something related to the demographic history or drift. And the other thing is this trait diving is a polygenic, or is just a single gene that is acting on this specific trait?
Okay, so in terms of the other signals, it's hard to say why those have been under selection. They could be false positives, but we did have a lot of different criteria for including a site, including it couldn't just be one point, it had to be a whole region. And so it could just be that there's a lot going on. I just think I have, oh no, I don't have it there. Anyway, so it could be that there are things that have been under selection for various other reasons that we don't understand yet. But I don't know. That's something that we're looking into. In terms of whether it's polygenic, that's a great question. I mean, there's a lot of really interesting studies going on right now with polygenic selection. I think that this is not the only thing that's making them better divers, but in terms of whether it's been selection, I mean, there's just so little known about even the heritability of spleen size, architecture, anything like that. It's really hard to say at this point. I think the fact that we see such a dramatic increase in the mouse spleens from just applying an inhibitor suggests that it could just be this one gene. But I think it's hard to say with the data that we have right now, but it's an interesting question.
Any other pressing questions before we go to tea? We will have more snacks and tea and informal discussion, which I encourage you all to come to. So with that, let's thank Dr. Illardo again.